Hello and welcome to today's webinar at Microscopy Focus, which is a Leica Microsystems production powered by Bitesize Bio. Today's presentation is titled Imaging GPCR Activity, Unraveling Mechanisms from Molecules to Morphology, and is being presented by Professor Aylin Heinialoglu and Anya Herdermani from Imperial College London. Professor Heinialoglu has been a group leader at Imperial College London since 2007 and a satellite group leader at the Francis Crick Institute since 2022. She received her BSc in Human Biology from King's College London and a PhD in Molecular Endocrinology from the University of Western Australia, undertaking postdoctoral training at the University of California, San Francisco. Her research focuses on mechanisms directing G-protein coupled receptor activity in diverse physiological/pathophysiological systems. Anya Herdermani is a final year PhD student at Imperial College London. Her research focuses on GLP-1 and PYY secreting enteroendocrine endocrine cells, or EECs, in the intestinal epithelium, and on the role of nutrient sensing GPCR in programming lineage-specific EEC differentiation. She is particularly interested in the use of confocal microscopy in illuminating diverse cell morphologies, as well as the intricacies of subcellular specific GPCR signaling. After the session, I'll be putting your questions to Aylin and Anya, so please type your questions as they come to you in the questions box, which appears on the top right of your screen. So now, over to you, Aylin and Anya, for the presentation.
Thank you so much for the introduction. So as it was said in the introduction, our group focuses our research on this superfamily of G-protein coupled receptors. And at the top of the slide, it's probably a bit of a biased statement, but the point I want to actually make here is how these receptors are central to all physiological systems. Now, I like to think of these receptors not just as sensors for an individual cell's environment, but how we as individuals respond to our environment. So when we're happy, stressed, in pain, when you're watching a movie, sitting down to your favorite meal, you're activating different G-protein coupled receptors. Also, there are a number of stimuli that we either consume or expose to that are ligands for these receptors, including a number of different drugs. So it's probably not that surprising that even small changes in the activity of these membrane receptors can lead to a number of different diseases, and I've just listed a few of them here. So these G-protein coupled receptors are 7 transmembrane domain receptors. So it's a single polypeptide chain that passes through the cell surface 7 times. Now, you and I have more than 800 different G-protein coupled receptors, and that share this kind of common architecture. Now, as many receptors as there are even more ligands. There are thousands of ligands, and they represent the most chemically diverse set of ligands, from photons of light to ions to a huge range of odorants that make up our sense of smell, a number of small molecules such as amino acids and small peptide hormones, and also a number of more complex glycoprotein hormones that I will talk about a bit more in a couple of slides. Now, with all these receptors and all these ligands, there are just these kind of 4 key G-protein pathways that they are activated, and these are heterochimeric G-proteins. And these G-protein pathways will activate their own effectors, and they are actually crucial for a number of fundamental cellular programs such as changes in metabolism, gene expression, hormone secretion, etc. Now, within our group, we are interested in unraveling the kind of molecular and cellular properties that regulate the signaling from these G-protein coupled receptors. And what to highlight here is that we are interested in a variety of different receptors, but primarily focused for those GPCRs important for female reproductive health, such as polycystic ovarian syndrome, in pregnancy, also including preterm labor. But we're also interested in GPCRs within nutrition and how our gut senses our food, with which Anya will talk about some of the research in this area. I also wanted to highlight that these G-protein coupled receptors are a known, successful, and highly tractable drug target, with currently around 34% of FDA-approved drugs will target a particular GPCR. So we are also interested in being able to translate the kind of molecular cell biology, not only into mechanisms of disease, but also could these be exploited as new therapeutic targets. So for this kind of first part of this talk, I'm going to be talking about a particular G protein coupled receptors that's really important within our reproductive axis. And this is the luteinizing hormone receptor, or LH receptor. And I'm calling this the amazing LH receptor. So for us and our group, it has been a really a prototype GPCR for some of the pathways that I'm going to be describing. So luteinizing hormone is produced from our anterior pituitary, and this is in response to release of another hormone, gonadotropin-releasing hormone, from the hypothalamus. Now, luteinizing hormone, along with its equally important related reproductive hormone, follicle-stimulating hormone, will act on its GPCRs within the testis and ovary. Now, for luteinizing hormone in the testis, this is really important in switching on testosterone production from the Leydig cells within the testis. And with the ovary, it has more wide-ranging roles, from androgen production from the theca cells to ovulation and release of the oocyte. And then once that oocyte has been released, keeping progesterone production from the corpus luteum. The other kind of point to highlight is this: for this G protein coupled receptor, the key signaling pathway, it's a GS coupled receptor, and what that means, it activates adenylates cyclases and increases the second messenger of cyclic AMP. But it will talk to other G proteins. It will talk to activate GQ11 signaling, and will increase second messengers, notably levels of intracellular calcium. But it will only do this under specific physiological conditions, when there is high enough hormone and high receptor. So our signaling models of G protein coupled receptors have really dramatically changed over the past 30 years. So we've really moved away from this kind of classical biochemical textbook model of a single receptor that sits on the plasma membrane, activates a single G protein pathway, and a very kind of linear downstream response occurs, to something that is much, much more complex. And the kind of mechanisms that we kind of have uncovered that kind of have contributed to this complexity is that we now know these receptors can talk to more than one G protein pathway. They can even activate signaling, other signaling pathways in addition to through its G proteins. That different receptor, or different ligands, I should say, to a single receptor may activate pathways differentially, leading to this biased agonism. There is an incredible amount of crosstalk that can happen across receptors, and this may be in part to these GPCRs being able to physically associate together as these homomers or heteromers. And we also know that the location of these GPCRs within the cell is really important for both diversifying the signaling and also specifying the signaling response. And this perhaps makes sense. If we have all of these GPCRs and all of these ligands, we have a very limited signal machinery, these kind of 4 key G protein pathways. These kind of mechanisms to diversify this signaling with this kind of limited amount of signaling machinery makes sense physiologically and potentially pathophysiologically. So the two kind of areas I want to kind of highlight here are our work that has kind of really, where imaging has elevated what we understand about the mechanisms of GPCR signal activity, and how we've applied imaging to understand how GPCRs associate together as homomers and heteromers, and also in our work in looking at membrane trafficking of GPCRs and its impact on signaling activity. So it was long thought that G protein coupled receptors within the plasma membrane exist as monomers. And now that we know these receptors not just operate in isolation, but they can associate with themselves as these dimers and even higher order oligomers. And this is thought of a way of potentially both amplifying signaling response or even diversifying its G protein signal activity. Now, this has been, I have to say, a controversial area, and this has been in part due to some of the tools that have been applied. And it's really important, if you are going to kind of delve into this, is to demonstrate that these kinds of associations are physiologically relevant. And the luteinizing hormone receptor was the first class A GPCR to show that these interactions were indeed physiologically necessary. The LH receptor has also been a great receptor to apply new imaging modalities to actually start to be able to directly look at how these receptors form these kind of complexes. And I'm going to be showing you some summaries of data for these two parts. So the work that was undertaken to show whether GPCR dimerization is physiologically relevant, as I mentioned, was work that was done with the luteinizing hormone receptor. And this was led by Ilpo Hutniemi, who used to be at Imperial College London. And what they use is a knockout model of LH receptor. And as you expect for something that's important in reproduction, if you knock out this receptor, these animals are infertile. And these are kind of data from male mice that are being summarized here. But to look at the question of whether dimerization is important, they created two different LH receptor mutants. So here we have the blind and the lame. So the blind receptor is a mutation in the receptor that cannot bind its hormones. You can imagine that this receptor is inactive. And if you express this mutant receptor, binding deficient mutant receptor, in the background of the knockout animals, they remain infertile. You take a second mutation, and this time our lame receptor, here we have a mutation within the transmembrane bundle. It can bind hormone but cannot signal, but it is a deficient receptor. And again, these animals are still infertile if you express it alone in these mice. But the question is, what happens if you put the two together? If these receptors then associate, you can have ligand binding on one of these receptors and then the signaling occurring from the other receptor if these two receptors are in a complex as a homodimer. And indeed, if you co-express these two receptors within the knockout background, you're able to rescue the fertility of these animals. So this was really important to demonstrate that these associations were physiologically necessary. But what our group wanted to do next was to actually be able to apply imaging tools to actually see this dimerization occurring. Was it a dimer? Was it more? Could there be a monomer? So there are a number of distinct single molecule imaging approaches that have been used within the field. And what I'd like to kind of point out with this is that you have different single molecule imaging modalities depending on the question that you're trying to address. So single molecule tracking approaches have been really great to uncover the association kinetics, the association and dissociation of two different molecules within the plasma membrane. But under these circumstances, in order to be able to resolve your single molecules, you have to underexpress or underlabel your particular target. Now, for our question, we wanted to get the full landscape of receptor complexes, and we wanted to be able to do this in physiological relevant conditions for our GPCR, the LH receptor, where we know that there, in the ovary in particular, there can be high receptor levels. So we turn to localization microscopy methods, specifically photoactivated localization microscopy, or PALM. Now, I have to say, while this allows us to be able to look at more denser samples, with techniques such as PALM, we do not have the temporal resolution that you would have with single molecule tracking. So there's always trade-off with these distinct techniques. So we have used a technique we called PD-PALM. So we use photoactivatable dyes, and we prefer to use photoactivatable dyes over photoactivatable proteins as they are brighter and more photostable. And we've used this PD-PALM technique now for a number of different GPCRs, for both looking at homomers and GPCR heteromers. And with this technique, we get to be around sort of 10 nanometer resolution. And simply the way that PD-Palm works, because we are using photoactive dyes, specifically cage dyes, they are in the dark state. So you need UV to uncage them before activating. And you only activate a very small subset of molecules, and then you immediately bleach those molecules. And then you repeat this process until you've activated and bleached all your molecules. And I have to point out that this is done within the TIRF imaging plane. So we are looking only at molecules at the plasma membrane. So I have to say, as someone who really loves imaging, I love microscopy. As images go, these PD-Palm images are not the most exciting to look at, because essentially what you end up with is lots and lots of spots. And then you have to work out what you're going to do with all of these lots and lots of spots. So what I've got here is just some example of the images for cells that are expressing those same mutants that I was talking about that was used in the mouse model. So the LH receptor binding and the LH receptor signaling deficient mutants, and cells that are expressing both. And we had to kind of design a software that would be able to count the monomers, the dimers, and the higher order oligomers. And the way that this software works is by a near neighborhood analysis-based method. So if we have one sort of molecule here, this is denoted here in 1, we can draw a specific radius around that molecule, and it will count the number of molecules within that specific radius. If it identifies another molecule, it will then take that particular molecule and draw the same radius around that without double counting, and it will keep continuing to do so. So what we're able to do with this is firstly be able to say something about how many of our molecules on the surface are associating together, and that's what's shown in the graph here. So whether it's our normal wild-type LH receptor or these two mutants, about 40% are associated, meaning that 60% are in the monomeric state. We can also get a further breakdown, so we can start to look at how many of these receptors are dimers, trimers, tetramers, pentamers, etc. And this was actually quite a surprise to us. So this is data taken in the basal state, so without being activated. These receptors are pre-associated in these very diverse set of complexes. When we look at cells that were co-expressing the binding and signaling deficient mutants, so these are ones that rescue the fertility in those animal models, then what we see is actually there seems to be a preference for these binding and signaling deficient mutants to associate with each other than on their own. The other nice thing that we can do with this PD-Palm imaging is to integrate that with molecular modeling. And this is work in collaboration with Francesca Finelli at the University of Modena. So we gave her a lot of different PD-Palm images. Specifically, we gave her lots of different trimers and little the tetramers that we were observing. And what she was able to do was to get a great match with her predictions, her molecular model predictions with our PD-Palm arrangements. The great thing with that is that Francesca is able then to see the rest of the receptor. So what we've got here, is our PD-Palm image, and this is kind of one of her predictions that matches that particular PD-Palm image. And here we've kind of assigned where our yellow and blue dots here, and here's the rest of the structure of the receptor. And this is kind of looking at sort of a top-down view. Then what Francesca is able to kind of look at is what the potential interfaces are, the dimerization or oligomerization interfaces are. And another thing was revealed is that the interfaces are equally as diverse as the kind of complexes that we observe. And this perhaps makes sense considering all the different arrangements that we are observing. So these kind of methodologies have further revealed the complexity of these associations. And we're now trying to uncover what this kind of can mean from a signaling viewpoint. And particularly, we're interested in looking at these complexes and within the ovary. So we've got ongoing work trying to look at this PD-Palm technology within ovarian cells to try to understand if these multiple complexes might have a role in all the multiple actions that LH would have in the ovary. Now, I just put a notice here that G protein coupled receptors can associate with completely different G protein coupled receptors, and we have done work in this space as other groups, but in the interest of time, I won't be discussing that today. So I then wanted to move on to our work where we have used microscopy as an absolutely fundamental tool to understand the spatial control of GPCR signaling. Now, classically, the role of membrane trafficking on receptor signaling is a way of regulating the responsiveness of a tissue to particular hormones simply by controlling the amount of receptors that are at the cell surface. And this is still an important role of membrane trafficking. But it also changes the pattern of receptor signaling. So a simple but dramatic example of this is the divergent sorting pathways that a GPCR can take once it's been activated and internalized into a cell into this early endosome compartment. Some GPCRs are rapidly recycled back up to the plasma membrane, where they can bind its ligand again and activate signaling, resulting in a more continued pattern of signaling, while other GPCRs are targeted to the lysosome and are degraded, resulting in a more transient signal response. Now, when we wrote this review back in 2008, we were talking about patterns of receptor signaling based on the idea that GPCR G protein signaling happens from the plasma membrane. But simply, it was only a year later when it was first identified that GPCR G protein signaling doesn't just happen from the plasma membrane, but either can continue or be reactivated once the receptor has been internalized into the endosome. And it was suggested that this second wave of signaling can result in a more temporally sustained response. Now, there's been a lot of activity in the past 14 years or so to understand what this means to a cell. So how would a cell decode a same G protein second messenger response from the plasma membrane, say versus an endomembrane? Could other GPCR G protein signal pathways be regulated in a similar manner? And what we're kind of starting to uncover is that there are potentially multiple compartments within the cell that GPCR G protein signaling can occur. So it's not only just from the plasma membrane or indeed the endomembrane. There are other cellular compartments that are reported, including the Golgi. Now, our contribution to this area has been to identify a new part of this spatial signaling atlas or map, and that is the identification of a new endosomal compartment. And the first clue that we had that there was such a new compartment was by comparing the internalization of two distinct G protein coupled receptors, the beta-2 adrenergic receptor and the luteinizing hormone receptor. Now, on the surface, these two GPCRs are similar in their signaling pathway. So they're both GS-coupled receptors, increasing cyclo-KMP. They internalize into the cell by similar mechanisms. They also rapidly recycle back up to the plasma membrane. But there are some very distinct differences when we start to look at the internalization. So these are HEK293 cells that are expressing each GPCR, and they are flag tabbed. So we've labeled the flag antibody. So here we have a single cell here labeled up. The receptor's labeled up here at the cell surface. And when I start to play the movie, when the timestamp appears is when the ligand for this receptor has been added. Okay, so in goes the ligand for beta-2 adrenergic receptor, and you'll see that the internalization is very robust. And you'll see that it internalizes into these what calls little, almost like little donuts. And these are the early endosomes. It's the receptors sitting on the limiting membrane of these endosomes. And if your eyes are very good, you can also sometimes see even the recycling tubules emanating from these structures. So if we do the same for the luteinizing hormone receptor, in goes LH. And you'll first see that the internalization appears within this particular plane of confocal imaging is not as robust as the beta-2 adrenergic receptor. But what really stood out was just the sheer size of these endosomes. They are much, much, much smaller than the endosomes that the beta-2 adrenergic receptor occupies. And they're about a third smaller for the LH receptor compared to this other GPCR. And I have to say that the Leica SB5 was absolutely critical in getting these kind of first stages of this project. So I'm going to summarize a lot of data into one cartoon of what we understand, what is going on with this specific small endosomal compartment. So here we have the beta-2 adrenergic receptor. We know that the signaling from the receptor primarily happens from the plasma membrane, but there is this smaller second wave of signaling that happens from the early endosome. We know that these kind of larger early endosomes have these kind of classic markers such as RAB5, EEA1, and they the primary lipid within this compartment is PI3 phosphate. For the LH receptor, these much, much smaller endosomes are also biochemically distinct. So they contain none of these markers. And what is also interesting is that the majority of the cyclic AMP for the LH receptor has to occur from these smaller endosomes rather than the plasma membrane. We know a little bit about some of the machinery that is involved. And the one that I really want to highlight is this protein here called APL1, which is an adaptor protein. And what APL1 plays a role in is to negatively regulate the signaling from this compartment. So it switches off the signaling, but also when phosphorylated by pKa to promote its recycling. So it's not just the LH receptor that uses this compartment, and we've named this the very early endosomal compartment, or VEE, just to distinguish it from the EE. So the FSH receptor, it's sort of LH receptor sister receptor, but also the beta-1 adrenergic receptor, the mouse short-chain fatty acid receptor FFAR2, and also a prostaglandin receptor EP2. Now, we are currently trying to unpick how this particular compartment operates. And what we really need to know is some of the machinery that's involved in regulating this compartment. So I won't be talking about today, but just say to watch out soon. We've been doing some proximity-dependent proteomics where we're hopefully able to report some new players within this compartment. But what this very early endosome has enabled us to do is to create a new model for GPCR signaling, at least for the subset of GPCRs, which we're hoping to potentially apply in specific diseases. But what I wanted kind of to touch on is how we've wanting to use this compartment to potentially see if we can pharmacologically modulate how the receptor signals just by tweaking how it engages with this very early endosomal compartment, especially given as GPCRs are obviously a highly tractable drug target. And to do this, we've actually switched away from the LH receptor to the FSH receptor, purely as there are a lot more small molecules available for the FSH receptor, as it is an active kind of target, particularly in the IVF field. And this is work that was done by Silvia Spazzini, and in collaboration with Tocopherex, who were able to provide us some distinct allosteric small molecule modulators of the FSH receptor. And we've provided with two distinct chemical series, this benzamide and this thiazolidone. And both of these small molecules, although allosteric, so they don't bind the same place as the native hormone, are agonists, and this is shown here within this dose response curve. But what was kind of really interesting is how they show very different properties when we start to look at the trafficking of these receptors. What I'm just going to show you here as an example of here is how the FSH receptor recycles on addition of its agonist. And the way that we measure recycling in the lab, it's a very rapid process. So we use this pH-sensitive GFP that's attached to the N-terminal extracellular domain of the receptor. And because it's pH sensitive, it will be only fluorescent at a pH greater than 6.5. But once the receptor is internalized within the more acidic environment of the endosome, this GFP fluorescence will be quenched. And this enables us, along with turf microscopy imaging, to measure the reinsertion of the receptor into the plasma membrane. When I play the movie, what you're going to be seeing is these, what looks like, obviously, we'll call them puffs. They're like these little explosions of the receptor into the plasma membrane as the receptor rapidly recycles following addition of FSH. But what one of these small allosteric molecule is able to do is able to induce this very dramatic increase and enhanced recycling capacity of this FSH receptor. And the reason that it does this is because it's lost its ability to be regulated by APOL1 for its cyclic-AMP signaling. So the cyclic-AMP signaling is elevated, we get more PK phosphorylation, and therefore we get more recycling of this receptor back up to the plasma membrane. So this small molecule, just by, it's a very small tweak in how it engages in this pathway, has resulted in enhanced activity. With the benzamide, the recycling isn't changed, but interestingly, it's lost its ability to be regulated by APOL1 for its recycling. So it's recycling by means that we have yet to be able to uncover, although its primarily signaling has maintained. So this has shown us that these molecules, just by very small changes within the very early endosome, has resulted in distinct changes in activity. Okay, so I think now we're going to have a slight change, and I'm going to pass you on to Anya.
Thank you so much, Aileen, and thank you for the introduction at the start. So my project is moving now to the space of the gut-brain axis. So I'm sure we've all heard of Ozempic or Wegovy, known as semaglutide. These are popular GLP-1 analogs at the moment. So GLP-1 is glucagon-like peptide 1, primarily known for its benefits as a glucose regulator in the body, and secondly, which is now gaining more therapeutic interest by the day, as an anti-obesity agent. And the way it does this is through what I mentioned, the gut-brain axis. So the gut-brain axis is a bidirectional means of communication that we all have, consisting of neural, endocrine, and paracrine pathways that essentially provide a means for your gut to communicate its nutritional environment to your brain, and your brain to action higher order functions, and conversely, your brain to communicate cues to your gut that might affect your digestive function. So particularly focusing on the release of appetite regulating hormones, such as glucagon-like peptide 1, we draw our attention to an enteroendocrine cell in the gut. So on the schematic in the left, you'll see something labeled as an intestinal L cell, which I'll explain in more detail in the next slide. But essentially, your gut contains a variety of nutrient-sensing cells with G protein coupled receptors on them that are able to respond to the food you eat and the metabolites that it's broken down into by secreting hormones such as GLP-1, another one being peptide YY, PYY, which I'll also explain later on in this talk. And these hormones are able to be transduced to your brain via your vagus nerve or other endocrine pathways, and essentially communicate with your hypothalamic brain centers as to whether you're full or you're hungry. This can also operate in hedonistic pathways, so things like cravings and voluntary food cues, which I don't have time to explain today. But I'm going to focus now on that enteroendocrine cell type in the gut. So like I mentioned, these enteroendocrine cells are gut hormone secreting cells. Collectively, although they only comprise 1% of your intestinal epithelium, they make up the largest endocrine organ in your body. So these endocrine cells are incredibly responsive to a variety of nutrients and nutrient-derived metabolites in our intestine. So the way they are able to exert this responsiveness is through a variety of G protein coupled receptors and nutrient sensing transporters. So for example, you've got the free fatty acid receptor family of FFAR1, 2, 3, and 4, which bind to a variety of fatty acids. You've got amino acid receptors, sugar binding receptors, and then of course, various transporters of sodium, glucose, so on and so forth. But essentially, by binding to these nutrients, these receptors, like Aileen mentioned, transduce a variety of intracellular pathways that are then transduced into calcium-dependent hormone excitosis in these cells. So I particularly focus on a family of GPCRs called the free fatty acid receptors, particularly members FFAR2 and FFAR3. So these are two free fatty acid receptors that are present on enteroendocrine cells known as L cells. Enteroendocrine cell subtypes, just briefly, are grouped based on early electron microscopy studies on them and their main hormonal products, which I won't go into for the sake of time, but intestinal L cells being the primary cells that secrete GLP-1 and PYY. So it's really interesting that receptors are expressed on these cell types, and their ligands are a group of molecules called short-chain fatty acids. Short-chain fatty acids are actually metabolites produced by your microbiome from dietary fiber, so by the anaerobic fermentation of dietary fiber that is resistant to digestion by enzymes of the human digestive system. So in vivo studies have shown that short-chain fatty acids are able to stimulate GLP-1 and PYY secretion from L cells via activation of FFAR2. But like Aileen mentioned, there's a whole complex diversification of signaling and downstream mediators that happens downstream of GPCR. So there's much to be learned and much to be uncovered in terms of understanding the mechanisms that lead to hormone release, and particularly given the huge anti-obesity interest on these appetite regulating hormones now, this is a really exciting therapeutic paradigm. So I'm going to introduce the model that I primarily used for this project. This cell line is called the NCI H716 cell line, derived from a colon adenocarcinoma. The cell line recapitulates many of the properties of endogenous L cells, including their nutrient responsiveness and their hormone secretory profiles. This cell line is dependent on culturing on extracellular matrix in order to fully recapitulate the endocrine properties of L cells in the gut. So I'm going to talk about some of the really exciting microscopy techniques that I've been able to achieve in this cell line in order to illuminate some of the really interesting functions of FFAR2 in driving GLP-1 and PYY hormone release. So as I mentioned in my introduction slide, short-chain fatty acids have been shown to drive the secretion of both GLP-1 and PYY, but there's emerging evidence that these hormones could in fact be regulated by extremely different mechanisms leading to their downstream release. So by use of the Leica Stellaris 8 and the adaptive deconvolution lightning module, we were able to clearly identify a separate localization of these two hormones within NCI cells into two very different pools. So the yellow line here indicative of a region of interest, and this is the intensity profile line plot of the two respective hormones, being GLP-1 in green and PYY in pink. And this is an analysis which was enabled by quantitative analysis of the lightning images, in which a Pearson's correlation coefficient was calculated to signify the overlap between these two hormones. As you can see here, it's very clear that these hormones co-localize extremely poorly, which was really interesting because lent itself to the question of FFAR2 exerting distinct mechanisms of regulation on these two pools. So just to take this a little bit further, we were able to visualize this adaptive deconvolution data using the LASIX software on the Leica Stellaris 8 in 3D. So here is the image I showed in the previous slide, but just in 3 dimensions on the Y, Z, and X axis, again, showing a really clear separation of these two pools of granules in these cells. So this really was a starting point for us to investigate how short-chain fatty acids might differentially affect these two hormones. So on the left-hand side, you'll see we explored the transcriptomic effects that these short-chain fatty acids have on these two hormones. And on the left-hand side in pink, you'll see that we saw a really clear, significant upregulation of PYY by these 3 short-chain fatty acids, C2 being acetate, C3 being propionate, and C4 being butyrate, which were the short-chain fatty acids in question due to their high potency at FFAR2. And on the right-hand side, in contrast, you'll see this downregulation of proglucagon, or the precursor of GLP-1. I will mention that short-chain fatty acids, in addition to activating FFAR2, are able to interact with the energetics and histone deacetylases of intestinal L cells, which has been the question of previous studies. So it was really important to clarify the role of FFAR2 in these effects. So it was really interesting to see that FFAR2 antagonism by a small molecule antagonist was able to diminish the effect of short-chain fatty acids on PYY. However, our preliminary data showed that on GLP-1, this inhibition wasn't as evident. So this really led us to further consider the possibility of FFAR2 really potentially only being involved in the regulation of PYY transcription, and GLP-1 perhaps transcription being regulated by a different mechanism. We were also able to visualize the butyrate mediated upregulation of PYY as a downstream increase in the secretion of the hormone by ELISA assay. So further downstream of FFAR2, we were interested in understanding the specific G protein pathways that might be involved in this upregulation of PYY downstream of butyrate treatment. So we employed 2 inhibitors of G proteins, pertussis toxin, or PTX, to inhibit GI, and YM, an inhibitor of GQ. So you'll see, indicated by the respective colors, that we saw a significant inhibition of the PYY transcriptory response with butyrate when cells were treated with pertussis toxin, but not with YM. So to further corroborate the involvement of a FFAR2 GI pathway, we used a selective FFAR2 GI ligand, AZ1729, and also saw an upregulation in PYY transcription independently, as well as a synergistic effect of butyrate's upregulation of PYY when this ligand was added in combination. So in order to understand the secretory mechanisms of PYY, particularly downstream of butyrate treatment, we employed a novel tool whereby we designed a fluorescent protein tagged construct of this hormone. So like Aileen mentioned, TURF microscopy really exploits the properties of an evanescent wave, which allows illumination selectively within 100 nanometers or so of the solid interface. So you're able to really zone in on events happening at the plasma membrane. And the pH sensitivity of this tag was such that we were able to detect a clear upregulation in the fluorescence upon vesicle fusion with the plasma membrane. As Aileen mentioned, within an endosome or a secretory vesicle here, you're looking at an acidic pH of 5.5. Upon exocytosis with the cellular membrane, the pH transitions to a more neutral pH, and this fluorescent protein tag is optimized to increase in fluorescence upon that transition. So upon cloning of this construct and transfection into L cells, we were able to detect an upregulation of these flash events following butyrate treatment, so further substantiating what we had observed by the ELISA assay in this cell line. So I'm going to play the video, and if you just look very carefully where this pink arrow is pointing, you'll see a fusion event characteristic of this assay. So there you go. So essentially, by combining TURF and the use of this construct, we were able to monitor PYY excitosis at single event resolution. So I just to illuminate another way we use microscopy to kind of uncover a novel effect of short-chain fatty acids on intestinal L cells. What was really interesting is that through our ligand treatments with short-chain fatty acids, we noticed these drastic morphological changes in cells, which initially was almost a mistake under a light microscope. We then were able to use a specific dye called cell mask, which stains the cytoplasm and the cellular membrane, to visualize these changes by confocal microscopy. So after taking these images, we were then provided this means for quantitative analysis of these changes we were seeing, and particularly if I can draw your attention to butyrate, and less so, but still propionate, you'll see a significant upregulation in the area and the ferret diameter, or also known as the longest distance across a cell, particularly with butyrate treatment, and less so, but still evident with the propionate treatment. So following on from these morphological changes, we sort of considered the possibility that these short-chain fatty acids were in fact stimulating cellular differentiation. So again, a lot of data on this slide, but what I will draw your attention to on the top half of the slide is your canonical differentiation cascade that happens in the intestinal epithelium, going from an LGR5+ stem cell, which are the bread and butter of most cellular populations in the intestine, all the way down to this mature L cell phenotype and genotype. So we particularly saw a significant effect on late stage markers within this cascade, so particularly NeuroD1 and PAX6, with butyrate again having this most significant effects compared to the other short chain fatty acids. We did see some other effects on early stage and intermediate stage markers such as 801 and ARCs, which were also relevant considering that expression of a lot of these factors are transient, and downregulation of these factors can be indicative of moving towards a later stage in the cascade. So finally, in order to, again, understand the role of FFAR2, the butyrate binding GPCR, we employed the small molecule agonist AZ1729 to understand that if this molecule was able to also recapitulate some of these effects we saw on differentiation. So really interestingly, we saw a significant upregulation of PAC6, the late stage differentiation marker, with the agonist independently, as well as a synergistic effect of AZ when treated with butyrate, further emphasizing the possibility that FFAR2 might serve this distinct role, not only in modulating the secretion of really therapeutic relevant appetite regulating hormones, but also actually in the differentiation of these cells. So I'm just going to pass on to Aileen finally to give a little summary of our talk today.
Thank you, Anya. So what we just kind of wanted to highlight is some take-home messages, is that for our research, that applying multiple imaging modalities have really shone a spotlight on new mechanisms for how these GPCRs work, not only in distinct cellular systems, but how these receptors could operate in distinct subcellular compartments. And what we're kind of really excited about is this is now providing us these new models to be able to work with, to kind of probe in and use in distinct diseases, as well as potential new therapeutic targets. So what our current challenge is as a group is to try and apply these imaging methods to more complex systems. I think there's a real need to kind of moving towards a system such as 3D cultures and organoids, and also to use patient-derived samples. But they, again, propose sort of challenges in themselves, not only to establish, but also to be able to use such imaging techniques. So we just wanted to end by both of us saying thank you to a lot of different people, including members of the group, a number of different collaborators, both at Imperial and other institutes, our funders, and to you all for your attention. Thank you.
Thanks both. It was a great presentation. It's interesting you say there are approximately 800 GPCRs, because it makes one wonder why that classical model of signaling ever arose. It's quite a few really for such a complex organism as us. And the study demonstrating the physiological relevance of the GPCR oligomerization, that was really elegant. So yeah, thanks, that was really nice. So we've got some questions from the audience coming in, so I'll try and direct these to the relevant persons. So Aileen, you mentioned a new model of spatial control of GPCR signaling that can be used to understand the mechanisms of disease. So what diseases are you currently applying this to, or would you like to apply it to?
Thanks, Tom. So one of the areas that we're really interested in is looking within polycystic ovarian syndrome. So this is a very common endocrine disorder. About, depending on the population, up to 15% of women of reproductive age have PCOS. And there is a lot of evidence to support the role of altered luteinizing hormone receptor signaling in this condition. And we have been trying to apply this model to try to understand how the receptor activity may be altered in these patient samples. So we're very fortunate to work with other groups at Imperial who are able to kind of access the ovarian granulosa cells from these patient groups. And the indications so far seem to suggest that how this receptor interacts with this very early endosomal pathway is very different. So you have this very enhanced activity of the LH receptor within these patient samples through how it's engaged with some of these proteins in this endosomal signaling system. So you have this kind of super active receptor as a result. So that's just one example where we're looking at the disease.
Sure, thank you. So you showed those models of the structural oligomers. Are you confident in those models? Have you got any confidence statistics in them? And have you attempted to validate them experimentally?
So we are currently doing that at the moment. So it is that those kind of models were based with aligning, so integrating the single molecule data with current structural data. And at the time we did this, we did not have the cryo-EM structure of the luteinizing hormone receptor as we do now. So we are reevaluating some of those models, but we're also looking at ways to try and disrupt the interfaces. So we've kind of created disrupting transmembrane peptide tools to start to see if these kind of complex interfaces can then, if we disrupt them, can we disrupt them, and what effect does that have on signaling? And we're doing that in cells, but also in more complex ovarian models as well, to kind of look at the physiology. So this is currently ongoing.
Sure, I guess it's so hard when they seem to form, so many higher order oligomers, and they're so hard to capture.
Really like to, it seems.
Proving their physiological order.
They're much more transient and probably easier to pull apart than this particular one.
Sure. So you mentioned that oligomerization could potentially impact signal amplification. Do you suspect there's any other physiological roles for that oligomerization?
Yes, so it could be one way to discriminate between different ligands. So for the LH receptor, there are two endogenous ligands between LH and the pregnancy hormone, which is human chorionic gonadotropin. And the way that the hormone communicates with the receptor to the signal machinery seems to be done in different ways. So we think that luteinizing hormone needs this kind of aura amplification, whereas HCG probably doesn't need these kind of complexes to form. So the directionality from ligand to receptor to G protein is kind of very different. So it's much more kind of linear through the molecule. So that could be one aspect of kind of ligand discrimination.
Nice, thank you. So I had a question come in. Someone's wondering, how can research integrate imaging data with other biochemical and biophysical approaches to gain a comprehensive understanding of GPCR function?
So I guess one example that was given was single molecule imaging data with molecular modeling, and that was really combining those two together that we've kind of really tried to understand how these complexes are formed. We wouldn't be able to have done it in any other ways. I mean, I guess routinely using, if you're looking at GPCR oligomers or dimers, then sort of biochemical and biophysical approaches may complement some of the data, but once we got into the single molecule side, realize the devil's in the detail, and what you kind of might see from associations with one technique is when you kind of start to drill in to what the individual receptors are up to, it starts to kind of uncover further mechanisms.
Sure, thank you. So I guess this one's for you, Anya. So given the hot topic nature of weight loss drugs such as Ozempic, how could the findings regarding butyrate be used in combating obesity?
Yeah, so it's a great question. I think it's just being caught onto in the pharmacological world, but the issue that patients are currently facing with GLP-1 analogs and similar products are the intense dosing regimens because of the effects not being prolonged. So in terms of understanding how GPCR ligands like butyrate might in fact have this effect on the tonic secretion of these hormones, if we can optimize that for patients, this might, through dietary approaches as well, because ultimately short chain fatty acids are metabolites of fiber. So if food regulations could have foods tailored to encompass more prebiotic properties and butyrate production, but also then thinking from an FFAR2 pharmacological side, if these ligands can just reprogram or induce a cellular phenotype to a more endocrine optimized form, then essentially your gut is working for you, instead of having to continuously take these analogs over time, and they're a repeat wearing off, and also the reversibility of the effect as soon as you stop taking a medicine like Ozempic.
Sure, thank you. So two very similar questions, so I'll ask them together, and I don't know who best to answer them, but how does the spatial and temporal resolution of imaging techniques impact our ability to study GPCR signaling dynamics? And as a follow-on, what are the current challenges or limitations you have in answering questions about GPCRs from an imaging perspective?
These are big questions.
Very broad.
Want to tackle? You want to start? Yeah.
I think a particular struggle that me and some of my colleagues have faced is the lack of available really specific antibodies for GPCR. So as cool as it is to visualize these GPCRs really easily in these transfected heterologous overexpression systems, ultimately it would be incredible to be able to really easily illuminate the spatial arrangement of these GPCRs on the cell surface. In the context of my project, it's unknown whether FFAR2 and several other GPCRs in enteroendocrine cells are on the apical or the basolateral side. And it's really important when thinking about the dosage and even the identity of ligands they're exposed to. So I think that has been a challenge yet to be addressed effectively. I don't know if you agree, Eileen.
Oh, completely. And just to kind of add to that, there is obviously a lot of commercially available antibodies, but it's really up to the researcher to characterize them, even though they're selling them. It does not mean that they are these
cheeky. Yeah. Okay, thank you. So thank you for the nice presentation. And they've asked, is it possible to show cyclic AMP production from endosomes by microscopy, and how would one differentiate that from cyclic AMP produced from surface receptors by imaging?
Yeah, absolutely. So I think there are different tools that people have used. That's some that we've used and some other groups as well. And they range of using sort of the cyclic AMP sensors that you can visualize, like EPAC-based sensors, FRET-based sensors that you can see where the cyclic AMP signaling is occurring from, or even actually anchoring them to specific compartments. So you can use other proteins to either anchor to the plasma membrane, anchor them to the endomembrane. So we've used nanobodies that recognize the active form of the G-alpha S protein that will activate cyclic AMP signaling. And that was a great tool to visualize the active receptor G protein within these very little endosomes. And that's where we saw the primary signal occurring from for those receptors that occupy that very the endosomal compartment.
Sure, thank you. So you mentioned Eileen, that this new very early endosomal compartment, and one big push within the group is trying to identify what other molecules are within it. I just wondered, has anybody tried to isolate it to try and identify what players are in it that way? Is that feasible, or do you think it's impossible? Is it too delicate, or is it still too early to say?
Well, yeah, no, I do get we asked that question quite a bit. It would be a great way to do it. And our view is that it would be almost that if we do, once we've got these kind of new players from the proteomics that we have done, is to use that way as a way to then isolate this compartment. I think this compartment is quite a heterogeneous compartment in itself. And if we just use the receptor alone, we know that the receptor will be in other locales as well. So it might be a challenge, but I think it's definitely worth trying, Tom.
Sure. So it's like you obviously need to characterize it a bit more, so you gain some leverage to ways that you could isolate it.
Yeah,
that makes sense. Okay. So lots more thanks coming in. And somebody's asked, how do you know if an internalized GPCR is recycled through resensitization or degraded? And is the APOL1 molecule the molecule that recycles all GPCRs, or are there other APOL1 homologs or other molecules altogether?
So there are different ways that you can look at the kind of post-endocytic sorting of a GPCR. And there are kind of biochemical ways with biotinylation and trying to track the receptor that way through degradation or even recycling. There are image-based ways, and one of them I presented using the pH-sensitive GFP to look at kind of very rapid recycling dynamics. I'm just looking at the question again. Sorry. So
You'd have to repeat the question, sorry. That's just about Apple one.
So Apple one is only a molecule that helps to recycle receptors for those receptors that enter this very early endosomal compartment. So the beta-2 adrenergic receptor, which will primarily traffic to the classic early endosome, will be kind of recycled via other proteins from that compartment. So you can knock down APL1, it has no impact on the activity of that receptor. So APL1 itself is not a marker for that compartment, the very endosome compartment, but the functionality is. So that's a way of discriminating for those receptors that need that very early endosomal compartment for its signaling and sorting.
Nice, thank you. And just one more question. I don't know if it'll mean anything to you. It doesn't immediately register with me, but someone's mentioning a technique called DNA paint as a promising method. Do you think it could be applied to GPCRs? I don't know if you've heard of that method.
Yeah. So it is something that we have, and it has been used in the GPCR field for looking at dimerization, oligomerization. We are in discussion with another group if it's something that we could potentially look at receptor G protein associations at the single molecule level as well, because there is kind of quite nice dynamics that you could potentially get with DNA paint as well. So absolutely yes, and there are kind of various kind of single molecule approaches depending on, like I said, for the question that an individual researcher has.
Okay, nice, thank you. And then last question, as far as I'm seeing, which ligand was used to internalize the beta-2 androgen receptor, the B2AR?
So this is isobutanol, and it's kind of commonly used ligand for looking at the beta-2 adrenergic receptor. It's not beta-2 selective. It obviously will activate the beta adrenergic receptors. It's a beta adrenergic receptor rather than a beta-2 adrenergic receptor, specific ligand.
Okay. Thank you. Yeah, that was great. So there's no more questions coming in. So I guess with that, we'll wrap up. So that brings us to the end of the webinar. Thanks again for a great presentation. Thanks to the audience for taking the time to listen in today. If any questions come in from now to the end of the presentation, we can address these offline. And until next time, goodbye from all of us at Leica Microsystems and Microscopy Focus.
Thank you. Thank you, everyone.
Yeah.